Good morning. This is Helen Meissner, Director of the Tobacco Regulatory Science Program. Welcome to the webinar. We'll be discussing two new RFAs for R21 grants, maximizing the scientific value of the existing biospecimen collections and also secondary analysis. For those of you who are new to this area of tobacco regulatory science, I'd just like to give you a little bit of background. This program emerged as a result of a partnership between the FDA Center for Tobacco Products and the NIH. The Tobacco Regulatory Science Program has oversight and responsibility for the trans-NIH activities that with the Center for Tobacco Products, so there are multiple NIH institutes that participate in this program, as well as the Center for Scientific Review that oversees the review of the applications. I do like to mention always that TRISP represents an addition to the NIH Tobacco Research Program and doesn't replace or diminish any existing tobacco research activities at the NIH. As you know, NIH has really decades of experience supporting tobacco research, and this program with the FDA is in addition. And if you find that your research doesn't quite fit in the tobacco regulatory science arena, it's quite possible and very likely that you would still be able to find a home at the NIH. So I encourage you to look at the TRISP website. All of the grants that have been funded through the program are archived on the website, so I think that, in addition to other information about some of the research priorities, should give you a pretty good sense of the type of applications we're looking for. So today's webinar will just have some very brief introductory presentations, and then most of the time will be left to questions and answers. The slides will be posted on the website, and if your question doesn't get answered today, or if you think of a question following this webinar, there are scientific contacts in both RFAs that you are encouraged to contact. So I will turn it over now to our CTP colleague, Dana Van Bemmel.
Good morning, everyone. I'm Dana Van Bemmel. I'm the branch chief for the Research and Knowledge Management branch at the Center for Tobacco Products Office of Science. Thanks for the invitation to be here today. I'm just going to spend the next 15 minutes or so talking to you a little bit about the Center for Tobacco Products and our research program for those of you that might be new to tobacco regulatory science, and give you a little, hopefully, some brief insight into how we use tobacco regulatory science in our regulatory activities at the center. So just briefly, I think many folks are familiar with the Center for Tobacco Products. It was established in 2009 upon the signing of the Tobacco Control Act, the Tobacco Act, the TCA. It initially set up FDA to regulate the manufacturing, marketing, and distribution of cigarette tobacco, roll-your-own, and smokeless tobacco products. That was expanded in what we call casually the deeming rule. It was finalized in August of 2016. That allowed FDA to expand that tobacco regulation to include products such as electronic devices, cigars, water pipes, and any future tobacco products that we may not have thought about at this time. The Tobacco Control Act gives CTP the authority to do a number of things, including pre-market review of new products and modified risk tobacco products. It allows for some post-market surveillance. It allows the center to set product standards, which we'll touch on in a few slides. It provides an opportunity for adverse event reporting related to tobacco product use. It includes activities around health warnings, advertising and promotion, and the center as a whole is funded through user fees from the tobacco industry. Although CTP has been established with a number of regulatory authorities, there are also a number of authorities which are outside of our regulatory authorities. So it's always good to touch on those, particularly for those that are new to tobacco regulatory science, because it's things that are very commonly used in tobacco control, but not necessarily tobacco regulatory activities. So some of those include things like setting tax rates for tobacco products, regulating the tobacco growing, setting indoor clean air policies is something that's outside of the center's authorities. So I'm not going to run through each one of these, but you can see there are several things that may be common to you if you are familiar or working in tobacco control that are outside of regulatory activities. The Center for Tobacco Products is different than other centers at the FDA that you may be familiar with. It cannot regulate tobacco products using the safe and effective standards that other FDA centers use, and that's because tobacco, we know, is inherently dangerous. And so we have to use, we have a slightly different approach. We use what we call the public health model, looking at regulatory actions that are based on looking at risks and benefits to the population as a whole, including both users and non-users of the tobacco products. And you're going to see this public health model touched on several times throughout the next few slides. So I've talked a little bit about the CTP's regulatory authorities. Some of the ways that we use those regulatory authorities or intend to use include understanding the regulated product as a whole. Again, our review activities that I noted earlier, including new products and modified risk products. We can restrict marketing and distribution to protect public health. We can enact policies, guidances, and rules to decrease the harms of tobacco products overall within those areas noted earlier. We have a program, the Office of Communication and Education, that is focused on educating the public and communicating tobacco regulatory science and tobacco activities related to tobacco products to the public, and we'll touch on that in a little bit. And then where our office focuses and what our branch primarily focuses on is the expansion of the science base for regulatory action and evaluation. So regulatory science is different than what we consider the traditional NIH model for research. Tobacco regulatory science tends to perhaps be not as innovative, not as novel. We don't necessarily have research that's focusing on the mechanistic activities of how something might cause a disease. Tobacco regulatory science is really used, it's really a discipline as a whole used to inform regulatory actions, rulemaking within all centers of FDA. And so here on this slide, I'm just trying to compare for you regulatory science and other centers at FDA compared to tobacco regulatory science. And in essence, it's really any research, any science that's informing our regulatory actions. We have a number of different disciplines that participate in the regulatory activities that I'm going to touch on in a few slides, including disciplines that look specifically at the products, such as chemistry and engineering, disciplines that look at the users of tobacco products, including pharmacology, addiction, product use behavior or social science, toxicology, and pharmacology. And then those that may look at the population as more as a whole, including environmental assessment, statistics branch, evaluation, epidemiology, and consumer perception. So really within the Office of Science, within the center as a whole, we have a number of different disciplines that participate in our regulatory activities and come together to discuss and pull together whatever activity we might be working on. So I've mentioned regulatory activities a number of times. I'm going to just take the next few slides to touch on a few examples of our regulatory activities in the hopes to give those of you that may be new to the Center for Tobacco Products or to tobacco regulatory science a sense of the kinds of activities we're doing and how your research might relate or might inform current activities or future activities. So the first one I'd like to talk our product standards. This is one example of our tobacco product regulation. Examples of potential product standards, really anything that we might, any rule or guidance that we might put in place to regulate things like nicotine yield in a tobacco product, or constituent content or exposure to that constituent through the smoke or vapor. Restrictions on sales and distribution can be considered a product standard, and form and content of labeling. So those might be slightly different than things that may be a little more obvious, like a nicotine content or construction or components. But all of these bullets represent examples of potential product standards that could be put in place that are within our regulatory authorities. When we think about the statutory questions that have to be answered in order to put a product standard in place, we're thinking about what that standard, how that standard is appropriate for the protection of public health. Again, looking at both the risks and benefits to the population as a whole. Some examples of activities just over the last couple of years that would fall under our product standard activities include the Notice of Proposed Rulemaking we published in 2017 related to a product standard for NNN. The Advanced Notice of Proposed Rulemakings, which were published in March of 2019, and those included potential product standard for nicotine levels in combusted cigarettes, the regulation of flavors, and regulation of premium cigars. And so within the Advanced Notice of Proposed Rulemaking and the Notice of Proposed Rulemaking, there were opportunities for comments. If you're not familiar with these activities or you'd like to learn more about other regulatory activities that are happening, I've included a link at the bottom of the slide that'll take you to the CTP website, where there's a list of all of our activities that have been published, I should say. So the second regulatory activity that I wanted to touch on briefly is product review. This is something that we spend a lot of time on within CTP and the Office of Science. I mentioned a few slides ago that product review includes pre-market review, modified risk review. It also includes review that we call substantial equivalence, which is really looking at a product to see whether the product under review is substantially equivalent to a predicate product that's already on the market, and investigational tobacco products are products that may be used in research. So again, some of the statutory questions that we think about when we're looking at these various reviews are listed here on this slide. When we look at pre-market, we are asking ourselves if the marketing of this new product is appropriate for the protection of public health. Modified risk product review includes looking at product applications and asking whether or not that product is actually used by the consumer to significantly reduce harm and risk of tobacco-related disease, and how that might benefit or cause risk to the population as a whole. And then substantial equivalence I mentioned earlier. Some research areas that help inform product review. Again, this is not all-inclusive. This is just a list to start to get you thinking about research areas that might be helpful to tobacco regulatory science. We look at materials, ingredients, and design of the product. We think about how the product's being used, including its appeal, its behavior, use, initiation, cessation, perception, and then overall public health, morbidity, and mortality. The final regulatory activity that I'd like to touch on just briefly is the public education campaign. Again, these activities are led outside of the Office of Science and the Office of Health Communication and Education. We have a number of different campaigns. First, I'd like to just touch on the youth campaigns. Some of these you may have seen. There are advertisements, they're online, they're on television, they're in your Instagram feeds, and other social media areas. So we had the Real Cost campaign, and then the Fresh Empire campaign, and This Free Life. And so really all of these were geared towards the different subpopulations of youth, trying to be messaging more specifically to their interests and how they may be communicating and receiving the message. But all of these were meant to be educating the campaign on educating these populations of youth on the tobacco risk. In 2018, we also released an adult campaign called Every Try Counts, and this campaign is focusing, it's still out, on increasing the motivation to quit smoking among adult tobacco users. And I didn't include a link on this, but if you're interested in the education campaigns, what might already be out there, some research that might be helpful in informing the evaluation of these campaigns, these are online on our Center for Tobacco Products website as well. And these areas of interest are outlined in the RFA. They're also on our website and expanded on, but these are the primary research areas of interest that we use to guide our entire research portfolio at the Center for Tobacco Products. And finally, I just wanted to leave you all with some examples of areas of interest, areas of research that may be of interest to the Center for Tobacco Products, areas that might help inform any of these various regulatory activities that we briefly touched on. These are just examples. But any kind of research that helps us understand the impact of changes to tobacco products and the characteristics on dependence or any type of use, initiation, cessation, switching, particularly among youth and young adults, is definitely of interest to the center and would be helpful in a number of different regulatory activities. Research that looks at understanding how product design characteristics might impact constituent exposure, including toxicity or potential toxicity or exposure to harmful or potentially harmful constituents would be of interest. Biomarker research, which obviously is tied to this RFA, that would help assess exposures as well as harm and toxicity of tobacco products, including newer tobacco products such as ENDS. Any research that might help identify misperceptions among users and non-users, and any research that might demonstrate new approaches to communication, tied again to some of the education campaigns that would help promote greater public understanding to the risks of tobacco products. You want to see it, somebody? Okay, so I think that's my 15 minutes. Thank you so much.
Thank you, Dana. We're going to hold the questions until the end. So next we'll be hearing from Rachel Grana-Main from the National Cancer Institute, who's the program official, and we'll be talking about the RFA on secondary analyses.
Thanks so much, Dr. Meisner. I appreciate the opportunity to participate in this.
Could you just hold up?
I really enjoy my role as the liaison for this program and talking to applicants, which hopefully some of you will be contacting me, and the other scientific contacts. So the purpose of this particular RFA is to encourage the analysis of public use data sets that can inform tobacco regulatory actions in the U.S., as you heard from Dr. Van Bemmel. But specifically, these R21 applications should propose innovative analyses of existing publicly available, nationally representative U.S. cross-sectional and longitudinal data. So importantly, you're using existing data sets in these applications and not new data sets to investigate new research questions that will inform and generate findings that are directly relevant to informing FDA's regulation of the manufacture, distribution, and marketing of tobacco products. Other publicly available data sets can be used, but priority is going to be given to nationally representative analyses. So if you do intend to use a non-nationally representative data set, you just need to justify why it's appropriate for informing these regulatory priorities. As Dr. Van Bemmel went over, there's certain scientific research domains that they're particularly interested in that are outlined on the TRISP website, as well as somewhat in the RFA. That's the knowledge, attitudes, perceptions, behaviors, dependence, toxicity, health effects, etc. We wanted to call to your attention from the RFA, there's example data sets given. You are not restricted to using these data sets, but it is encouraged to pursue new research questions that can be used to generate these nationally representative findings from these particular surveys, for instance. Some of them are exclusively with youth. Some have biospecimen collection. and that will be important for which kind of research question you're pursuing. I wanted to include this link on additional data repository at NIH for your interest and perusal. It's important to acknowledge that when you're using these existing data sets, you may want to answer a particular research question. It may be beneficial and optimal to merge or link existing data sets. For instance, data that's collected at the person level can be linked with some of these nationally representative data sets that give policy information or geographic characteristics. One example that was given, again, this is not the only example we're interested in, but you could be linking the tobacco use supplement to the current population survey, which provides individual level data, to other CPS supplement data sets that could provide other levels of information. We're really interested in innovative analyses of extant data, as I was mentioning at the beginning. So with these applications, you should be pursuing new aims that are not being addressed with existing data set and existing data collection. That capitalize on the existing data collection. New advanced methods is also of interest, and like I was mentioning, the novel combination and integration of data sets. Importantly, research using the extant data that analyzes effects or outcomes that were not previously examined in the original scope of research for that data set is a priority for this announcement. I also wanted to highlight here, I won't read everything, but the application should not propose to carry out the currently ongoing data analysis or maintenance and distribution of those data sets. So in the RFA, which I encourage you to read closely, there are priority research questions listed, and that are included here, but they're not limited to these. If there's, you have a novel idea, of course, and identify a research gap that you think is very important to the FDA Center for Tobacco Products, that's encouraged as well. But these are priorities that have already been identified. And I will go over them briefly, and please read them in depth yourself. Identifying between-person and within-person changes in tobacco use patterns. Patterns and trajectories of tobacco product use are priority, as well as investigating the role of electronic nicotine delivery systems such as e-cigarettes in the initiation and use of other tobacco products and the influence of those product characteristics on those patterns and trajectories. Identifying patterns associated with cigarette switching, as well as changes in time, like I was mentioning the trajectories research, but also related to biomarkers among and within population subgroups. Product research, examining the effect of different quantities and counts of tobacco products, like how many cigars or cigarettes are offered in a package and how that influences use patterns. The role and influence of flavors in the patterns of tobacco product use, including ends. Risk perceptions, as was mentioned, marketing, packaging, labeling, and how that influences tobacco use patterns and behaviors among important subgroups. Health outcomes from the various tobacco products, except cigarettes. This RFA is not calling for further research on the health impact of cigarettes. The long and short, by itself. The long and short-term health effects of transitioning, comparing biomarker expression profiles with self-reported use, so using this data along with biomarker data. And further details about this can be found on the TRIS website. So as also mentioned in the RFA, I wanted to highlight that these applications can focus on only one class of tobacco products, such as the range of cigars or ends, or it can examine research questions that pertain across classes of tobacco products. If you are using or plan to employ analyses to investigate these impacts in vulnerable populations, I wanted to highlight which vulnerable populations have been specified as important and priority, using young adults by race, ethnicity, low SES, rural populations, people with comorbidities such as mental health and other substance use disorders, military, LGBTQ, and pregnant women and women of reproductive age. Now, also specified in the RFA that is really important to pay attention to is that there are certain areas that may be responsive as in general to FDA CTP's regulatory authorities, like what Dr. Van Bemmel was mentioning, but for this RFA, we are not calling for that research, and these will be considered not responsive. Applications, obviously, that do not use existing data, since this is secondary data analysis. Applications that do not focus on tobacco products. So if you want to have a primary focus on marijuana or something else that is not responsive to this RFA, the graphic health warnings for cigarette packages, I'm not talking about non-cigarette products, but for cigarette warnings, that is not being called for in this RFA. And communicating harmful, potentially harmful constituents to the public is not in scope for this one, as well as the impact of marketing restrictions on adults. On other populations, it may be considered responsive, but not just to adults alone. And then studies of the demographics or risk perceptions that describe only exposure to advertising without linking that exposure to tobacco use behavior. And in addition, there's a few other really quickly considerations that have been mentioned in this RFA that I'll call your attention to, that even though it's secondary or exit data analysis, the research project should continue to meet the ethical conditions described in the RFA. And also this RFA is adopting the National Advisory Council on Drug Abuse guidance regarding tobacco industry funding of applicants that respond to this FOA, and I encourage you to read that guidance. So I am one of the scientific research contacts at NCI, along with some of my colleagues across NIH. Dr. Postow at NHLBI, Dr. Bautista at NIAAA, Dr. Kautz at NIDA, and Dr. Tyson at NIEHS. Depending on the focus of your proposal, you'll want to contact one or more of us. A letter of intent is not required, but it's very strongly recommended. This helps us evaluate whether your proposal is within scope of both the regulatory authority and the RFA. It's due 60 days prior to the application due date that you're responding to. Here's the information that you should include, and I want to put a plug in for sending us some draft aims if you feel comfortable, because those really help us evaluate your research questions and whether they're responsive, as well as the scientific context to give you scientific feedback. And just I'll leave you with one helpful hint from doing this over time the past few years. We really absolutely never want somebody to get to the stage where they're putting in a proposal and then the research is deemed not responsive and you've gone through all that work. So I strongly recommend that you discuss your research and these aims with your scientific research contacts from one or more of the institutes, people I just mentioned, including myself. This is particularly important for this RFA if you're using a data set that's not specified and you're unsure whether that's a priority. And then I encourage you to also name the scientific areas of interest in your proposed research so that it's clear what domain and what priority question you're answering, and submit and do all this process early to allow time, since there's many of us that will be responding and we have to coordinate. And for more information, you have all these resources, and I appreciate the time, and I will pass it along. Thanks.
Thank you. Next, and we're going to have to switch over the presenter mode, I believe. We'll hear from Dr. Lisa Postow at NHLBI. She'll be giving you some details about the RFA for maximizing the scientific value of existing biospecimen collections. Lisa, does she have control now of the slides? Okay, Lisa, I think you can begin.
Okay, can everybody hear me?
Yes.
Okay, great. So thank you, everybody. So my name is Lisa Postow. I am a program director in the Division of Lung Diseases, and I am the liaison from the National Heart, Lung, and Blood Institute to the Tobacco Regulatory Science Program. So I'm going to be talking to you about a related FOA to the one that Dr. Grana just talked to you about, but this one is maximizing the scientific value of existing biospecimen collections. Again, this is an R21. Do I have control over this?
Yes. Top of the screen, above your presentation, the small arrow pointing to your right.
Got it. Okay, so the purpose of this RFA is to stimulate exploratory research relevant to the mission of the Center for Tobacco Products using existing publicly available biospecimens currently stored in repositories in the U.S. An additional purpose is to maximize the scientific value of these stored collections and to provide researchers with an opportunity to generate preliminary data for subsequent research proposals. again, another purpose, just to reiterate, is to generate data for use in subsequent grant applications. So this is an R21. The purpose is to generate preliminary data. The research projects that are proposed must address research priorities related to the regulatory authority of the FDA Center for Tobacco Products, as described by Dr. Van Bemmel. So some biospecimen examples that are described in the RFA are listed here. This is not an exhaustive list, obviously, but these are some examples that we wanted to bring to the attention of the applicants. So the first is the Population Assessment Tobacco and Health PATH study. There are biospecimen collections associated with that. In addition, the National Health and Nutrition Examination Survey, NHANES, the Prostate, Lung, Colorectal, and Ovarian Cancer Screening Trial, PLCO, and the National Heart, Lung, and Blood Institute Biospecimen and Data Repository Information Coordinating Center, BioLink, has an associated biorepository. Now, all of these studies, the websites at which you can find information on the biospecimen collections that are held for those studies can be found at the link, as well as information on how to request access to those biospecimens and what process needs to be gone through for that. In addition, other biospecimen collections can be used, and I will get to that in a minute. So some relevant requirements for this FOA, for the biospecimens: the proposal must propose to conduct at least one new lab measurement, but that lab measurement data can be combined with pre-existing lab measurement data if that exists already. The collections do not need to be nationally representative, although those collections will receive priority. Now, there are cases, particularly the collections that are contained in the NHLBI BioLink. Many of those collections are not nationally representative, but they do contain other data, such as clinical data, that may not be contained in the nationally representative collections. So if you are proposing to use a non-nationally representative collection, the application must include why this collection is unique and why it contains elements that are not available in a nationally representative data set. So the request for the biospecimens from the collection can be initiated at any time, but it must be at least 30 days prior to the application due date, and your grant application must include confirmation of that, and there's more details for that requirement that are in the FOA. Biospecimens can be pooled with biospecimens from another collection if that's necessary to, for example, get a high enough number of biospecimens that are being analyzed, but this pooling should be justified in the application. So research must propose the applications must propose research that is relevant to one or more of these areas, and these are described in the frequently asked questions page, which I highly recommend that you read in detail in addition to the RFA. So I'm just going to briefly go over these, but they're described in more detail in the frequently asked questions page. So one or more of these must be addressed in the application. Compare biomarkers of tobacco exposure and potential harm related to use of non-cigarette tobacco products across surveys. You may identify and develop biomarkers of harm and exposure unique to new and emerging non-cigarette tobacco products. That includes both tobacco-specific and flavor-specific, including but not limited to e-cigarettes. You may examine the effect of chronic exposure to water pipe tobacco smoke or hookah on biomarkers of inflammation and other harm. You may utilize urinary specimens to analyze biomarkers of tobacco exposure not currently measured by other national studies in urine. You may utilize blood specimens to analyze biomarkers of tobacco exposure not currently measured by other national studies in blood. You may examine alterations in genetic and epigenetic markers associated with exposure to tobacco products. You may examine the health effects of transitioning from combusted to non-combusted tobacco use using biomarker outcomes. You may prospectively compare biomarker expression profiles to self-reported tobacco product use and analyze measures associated with biomarker of exposure that may inform risk associated with tobacco exposure for mortality, morbidity by age, sex, and race ethnicity. You may assess between-person differences and within-person changes over time in attitudes, behaviors, exposures to tobacco products, and related biomarkers among and within population subgroups or by risk factors. And finally, you may identify and examine biomarkers of harm and exposure between dual and polytobacco users, including but not limited to electronic nicotine delivery systems and combusted tobacco products. So again, your application must address one or more of these. You do not have, it will not be more highly regarded if you address more than one of these, but you must address at least one. And all aims, again, must fall within CTP's regulatory authority, or else the application will be deemed non-responsive. So non-responsive research topics, again, this FOA also has areas of research that may be responsive to normal CTP regulatory authority questions, but they are not responsive to this particular RFA. So non-responsive applications would be those that do not use existing data or biospecimens, those that focus on non-tobacco products, short-term studies of the acute effects of reduced nicotine content cigarettes, studies of short-term health effects and or acute topography, clinical pharmacology testing of early generation ENDS products, and again, any other application that is outside the regulatory authority of CTP. So like Rachel, I am going to strongly encourage everybody to speak to the relevant scientific contact or contacts on the RFA. Again, these are the same scientific contacts for the secondary data analysis. R21 RFA, Rachel Grana-Main, myself, Lisa Postow, Dr. Bautista, Dr. Kautz, and Dr. Tyson. Like for the other RFA, letters of intent are due 60 days prior to the application submission due date, so these are the relevant dates here. And letters of intent should include this information that's listed here: number and title for the funding opportunity, descriptive title, information on the PDs, PIs, information on other key personnel, participating institutions, and again, I'm going to strongly recommend that the letters of intent include a draft of your specific aims so that we can review them prior to your submission to make sure that everything is responsive and we don't need to withdraw the application after it's been submitted, because again, that's a big waste of time for everybody, and we don't want that to happen. So these letters of intent should be sent to the TRIS mailbox that is listed here. These are the due dates for the applications. There are 3 due dates for this FOA. Budgets, this is an R21, so budgets are limited here. Budgets are limited to $200,000 in direct costs for any 1 year and $275,000 in total direct costs for the entire project period. And that entire project period is up to 2 years in length. And for more information, again, I highly recommend that you contact the scientific contacts that are listed in the RFA. You can also go to the TRIS mailbox, and I also, again, highly recommend that you take a look at the frequently asked questions page, as well as reading in detail the RFA. So thank you very much.
Thank you, Dr. Postow. I'd like to reiterate about the frequently asked questions that you might want to check that document from time to time, because we do update them when new questions come in. So it's a good idea to check it periodically. So now we're going to hear about the review process for these two RFAs, and I'm going to turn it over to Mina Anas, who's the Scientific Review Officer in the Center for Scientific Review.
Right. Okay, good morning, everyone. I'm Mina Anas. I'm a Scientific Review Officer in the Center for Scientific Review, and I will go over the general review principles involved in evaluating these applications and how the whole process is conducted and scored. And Center for Scientific Review, or CSR, is an arm of the NIH which will conduct the review of the applications received in response to both of these RFAs, 19021 and 19022. Now, some of this information the previous speaker has already told you, the letter of intent, the application due dates, there's one each in 19, 20, and 21. The scientific merit review will be approximately 6 months from the date the applications are due, and the advisory councils will review them for funding, again, approximately a few months later, and then the earliest start dates are also given here, either July 20, April 21, or September 21. So I'll give you the general peer review principles that is applicable to all NIH applications, submitted to NIH, and also specific review principles to these specific RFAs. So they will be reviewed and evaluated for both scientific and technical merits by a scientific review group convened by the Center for Scientific Review, and there will be two scientific review groups convened, one for the RFA 19021 and one for the 19022. And any person of conflict with the applications will be excluded. These will be people who are collaborating with them or who have any kind of interest in the study, and they will all be excluded from the review process so that it's an unbiased review. And the reviewers will have the relevant research expertise, and there may be some who could be federal employees of either the VA or the CDC. And the standard NIH review procedures will be used, which I'm going to show you in the next few slides. And every applicant, irrespective of whether the application is discussed or not, will receive a written feedback, which is typically called as the summary statement, listing all the evaluations. So this is an R21, which is a shorter grant compared to an R01, which goes over a span of 5 years. This is only for 2 years, as the previous speakers have mentioned. And what is typically, this is called a high-risk, high-reward mechanism. And what is specifically important for these kinds of applications is they're looking for novel scientific ideas or new model system tools or technologies. And you don't need to have an extensive background or preliminary information. And the emphasis will be on the conceptual framework, the level of innovation, and the potential to significantly advance our knowledge or understanding. And preliminary data are not required, although it may be provided if available so that the reviewers can evaluate the feasibility of the study. So there are 6 individual criteria, and there is an overall impact criteria, and I'm going to tell you in the next few slides what they mean. Excuse me. Overall impact is the score. So all these criteria are scored between scores of 1 and 9, 1 being the highest and best, and 9 being the worst. So in the overall impact, the reviewers will give an overall priority score that is anywhere between 1 and 9. That will reflect the assessment of the likelihood that the project will exert a sustained and powerful influence on the research fields. And that is a combination of the criteria that I'm going to mention in the following 5 slides, and then additional review criteria. So I'm going to now list the 5 criteria. The first one is the significance. Under this, what the reviewers will evaluate is, does the project address an important issue or a critical barrier, an issue or a critical barrier in the field? And if the aims of the project are achieved, how will scientific knowledge and technical capability will be improved? And how will the successful completion of these aims affect the concepts, methods, and technologies related to the manufacture, distribution, and marketing of the tobacco products? So that's what is evaluated under significance. So again, this will be scored between 1 and 9. And then the next is the investigators. So are the program directors, or the PIs, or the collaborators, and other researchers, are they well suited to the project? And if they're early stage investigators, or new investigators, or during the early stages of independent carriers, or do they have the appropriate experience and training? And if they are established, have they demonstrated an ongoing research accomplishment that have advanced their field? And if it is, you can also submit projects where there are multiple, principal investigators or MPI, multi-PI projects, and under that, they will evaluate whether the investigators have complementary and integrated expertise, and whether their leadership approach, governance, and organizational structure, whether they are appropriate project to also be evaluated. And the next criterion is innovation. Does the application, under this, they will evaluate whether the application challenges and seeks to shift the current research in the field of tobacco science as it relates to the manufacture, distribution, and marketing of tobacco products. And is this a refinement or an improvement or a new application of theoretical concepts, approaches, or methodologies, or instrumentation? So it can be either conceptual innovation or a technical innovation. And will the outcomes of the project provide new information to further develop knowledge base that informs the manufacture, distribution, and marketing of tobacco products in order to protect public health? And then the next criteria is approach. Under this, what they will evaluate is whether the overall strategy, methodology, and the analysis are the well-reasoned or appropriate to accomplish the specific aims of the project, or potential problems, alternative strategies, and benchmarks for success presented. And if the project is in the early stages of development, will the strategy establish feasibility and will particularly risky aspects be managed? That's what they will look for. And continuing on that, if the project involves clinical research, are there plans for protecting human samples from research risks? And are there inclusion of minorities and members of both sexes and genders, as well as the inclusion of children as justified in terms of the scientific goals and research strategy proposed? And they will also evaluate the environment in which the research is conducted, the institution or the university. Under this, they will look for whether the scientific environment in which the work will be done, will it contribute to the probability of success? And is there institutional support? Is there equipment and other physical resources available to the investigators that are adequate for the project proposed? And also, will the project benefit from the unique features of the scientific environment and the subject populations or collaborative arrangements, as the case may be? And these are also additional review criteria that they look for if it involves human subjects, whether there is protection of human subjects, whether the inclusion of women, minorities, and children, and vertebrate animals probably may not be applicable, and also biohazards, if there is enough description of what kind of biohazards are being used and if there is enough protection for the people involved. These are also will be evaluated, and these are scorable. And then, as I said before, the reviewers will use the NIH scale of 1 to 9 in integers, with 1 being the best and with the highest priority, and 10 being the worst. And all the eligible reviewers who are at the meeting, at the panel, will vote, and the average score is multiplied by 10, and then it is rounded to the nearest integer. So the average score will be in decimals because it's an average of all the scores, and that will result in impact scores of 10 to 90. This is the overall impact, and as I said before, 10 being the best overall score. And as is typical in all NIH applications, only the applications that are the highest scientific and technical merit, typically the top half, or only 50% of the applications will be discussed, the others will not be discussed. And those will be assigned an overall impact score based on the voting during the meeting. And there are also criteria that are not scorable. The reviewers will comment on these factors, but they will not factor into the scores. And these include select agents, things like, chemicals that are not under the biohazard category, and then resource sharing plans. If the project generates data or their plans to share it with the other scientific community, and they will also evaluate budget and the period of support, although there is a limitation in terms of the R21 application, as the previous speaker has mentioned. And also, we will also look at whether foreign applications or involved, foreign scientists are involved, and if they are relevant to the U.S. population and regulation. And then these are some additional criteria that are specific to the FOA 19021, And under this, they will also evaluate whether the variables are appropriate for the specific aims and whether the types and numbers and volumes are justified, and is there validation, accuracy, and robustness of the methods that are being proposed to analyze the biomarkers, and then are the effect sizes are sufficient, are there enough power calculations, and also do they have the power levels that, do they have enough samples to achieve the power levels that they want to achieve. And also they will look at statistical analysis that are germane to this particular analysis under this FOA. And finally, like the previous speaker mentioned, there are also some projects that will be excluded. This is based on this in that anything that does not use existing specimens, although there is they will be accepted appropriate justification if they are nationally representative. And then again, focusing on non-product tobacco products will not be included, and studies on short-term effects of reduced nicotine content cigarettes, and also short-term health effects, acute topography, clinical pharmacology, and testing of the early generation ENDS products. And then here are the review contacts. Dr. Ivins, both the names and all the information is available on the Center for Scientific Review website. For 021, Dr. Ivins is your contact for review, and then for the 022, Dr. Delia Olukomi-san is your contact. With that, I will end my presentation.
Thank you. We have one last presentation. That's from Amy Buckheimer, a Grants Management Specialist at NIDA, and she'll be addressing administrative and budgetary requirements. Once Amy is finished, we will open it up for questions. Amy, I think you have control now.
I hope I do. Good morning, all. Happy to be here, and I'm going to just briefly touch on administrative and budgetary requirements for these two RFAs. The NIH, from support from the FDA, will fund up to 7 R21s for the 021 RFA and up to 5 R21s for the 022 RFA. I'll just quickly go through this so that I can allow time for everybody else to ask questions. Clinical trials are not allowed with these RFAs. I will not read all of the letter of intent due dates, as those have been touched upon by previous presenters, but I do have all of the letter of intent due dates, the application due dates, as well as the earliest start dates for each, individually listed per slide. Project periods for R21s, as most know, are maximum of 2 budget years. And an item that I didn't include in the presentation, but I'd like to mention here, the RFAs, each of them, indicate that no more than $200,000 in direct costs may be requested per budget year, and also direct costs for the entire project period for both budget years cannot exceed $275,000, and those are just direct costs. Post-award information, mid-period progress reporting is due every 6 months for grants funded with FDA money following the project budget start dates. And should your grant be funded, you will hear from me, and I'll provide additional guidelines and instructions so that you know how to proceed. Mid-period progress reporting, incidentally, is also required if the grant goes into a no-cost extension. Finally, all FDA-funded awards are excluded from carryover authority. Therefore, you must submit prior approval requests, and I'll also provide additional instructions should the time come for that prior approval process. However, I would like to note that these awards, even though they're excluded from carryover authority, you typically can process your own no-cost extension for the first time via ERA Commons without prior approval. And I know it was brief, but I just wanted to get all that information in. If you have any questions or concerns, you can feel free to contact me. Thank you.
Thank you, Amy. Now we will open it up for questions, and as Alicia explained to you at the beginning of this webinar, you can chat and use the chat function in the on your computer to input the questions. So, Alicia, do we have any questions?
Yes.
Okay, do you want to read it?
Yes. We have one question, and this question is directed to Rachel Granamain. Are restricted use as well as public use data sets appropriate?
Yes, you need to apply for the restricted use files, but they're absolutely appropriate. For instance, with the past data set, the population assessment of tobacco and health, you need to apply and justify and go through a process to obtain the restricted use file and for public use, but it is encouraged and usually pretty good process now, for instance.
Any other questions? All right, well, we'll leave it open for a few more minutes. Okay. Okay, I just, as a reminder, this webinar is being recorded, so you will be able to go back to it, to the slides and the discussion. But we'll leave the line open a little bit longer to see if there are any additional questions.
So this is a question to all. What is the process for foreign institutions from outside the U.S.?
I'm not sure I know. Do you mean the application process? The question is a little bit...
We'll ask you to please restate your question for clarification.
Yeah, as someone just pointed out, I mean, in the FAQs, I don't know if this was answers your question, but one question that we had from an investigator was, could researchers include biospecimens collected in other countries in their proposal? And the answer to that is that foreign biospecimens may be included only if a data set is relevant to the U.S. population and U.S. regulation of tobacco. Applications not using nationally representative biospecimens will need to provide justification why the data set is unique and why the research questions cannot be answered from biospecimens collected in the United States. Yeah, and the same applies for the other RFA on data sets. I think it was mentioned in the presentation that it needed to be relevant to regulation in the United States. But foreign applicants are allowed to apply for this, both FOAs, but as mentioned, they still have the same requirements in that the research being proposed needs to be relevant to tobacco regulation in the United States. Thanks, Mary. So I don't know, is there a clarification of the question? Yeah, so the question is, are foreign institutions eligible to apply? Yes. Are there specific requirements that they should consider that were not mentioned in the webinar? I'm not aware of any other requirements other than the research proposed needs to be responsive to the RFA and relevant to the context of tobacco regulation in the United States. That said, if you have data that would be informative, then I think that would be fine. And I would recommend before applying that you do contact one of the scientists listed in the RFA to answer questions just to make sure that you meet the eligibility criteria. Is there another question? Okay, so another question came in for 022. If 5 awards are given for the October 8 deadline, will there be funds available for the remaining due dates? Yes, the answer is yes. That is for the fiscal year. So that I believe in the RFA, it says that's up to the number of awards allotted for fiscal 20. So yeah, there'll be funds available for future years. Would you give an example of proposals that would be outside of CPP's regulatory authority?
There's some on the FAQs, I think.
Right.
Mm-hmm. Go ahead. Did you have a...
I mean, we can give one that you kind of already said.
Go ahead.
But, if you're proposing to examine a research question, for instance, about the effect of smoke-free policies on behavior, that would be, as a primary investigation, that would be non-responsive, for instance, because FDA does not have the tobacco regulatory authority over instituting and enacting and enforcing smoke-free laws, for instance. So that's one that was already alluded to that we could call out. But
I think the other two that I know are on the slide that I may not have mentioned were related to setting taxes, related to tobacco product sales, and age restrictions. Those are both outside of FDA's regulatory authorities.
And I encourage you again to look at the frequently asked questions because there are quite a few examples there of different questions that would be considered non-responsive. And
the TCA.
To the Tobacco Control Act and to the RFA. And if you don't feel that they address your particular question, again, please contact one of the scientists listed in either RFA, and I'm sure that they will be able to give you a final weigh-in on whether your topic is responsive. So we'll wait another minute or two. I don't think we have any additional questions. I hope you found the webinar informative, and as we've mentioned, there are quite a few resources available to potential applicants. So we encourage you not to be shy and to get in touch if you have any further questions or interest in either one of these RFAs. So I think with that, we will conclude the webinar. Thank you for joining.